of Digital Kazakhstan, and we're here on Saturday, right after the Nadi's break, to commit ourselves to our ongoing seminar series as part of our Center of Competencies on Digital Science. Now, I do want to mention something important. You heard in one of the early presentations the significance of technology in many fields and the impact that big data and data analytics are having across many disciplines. But I think today's talk, more than many, will bring this point home. Because in my view, there's no field that will be transformed more substantially than the fields of medical care, health care, personalized medicine. We'll hear a lot about this today, but I'm not the only person to think so. I can quote authoritative sources, and I happen to agree with the assessment of Mr. Jobs that this intersection of biology and technology will be something profound, something transformative. So for us, this is indicative. In fact, an indication of this transformation is that in our own School of Science and Technology at Nazarbayev University, we're moving gradually from so-called wet labs, the emphasis on the classic biological laboratory setting, and we're introducing with each year and with each generation of students, we're introducing more dry labs, or what we might otherwise call computational facilities. And only 7 years ago, when we constructed our biology department, the department had no computational facilities. They weren't envisioned. And now a substantial proportion of the courses in that field are taught in computing labs. And the work they do in the laboratories is integrated very closely with computational resources. So this is why we focus so much in this session on what we call bioinformatics. But in fact, we should see this as a manifestation of data science in the field of biology that covers a broad cross-section of other disciplines. And I would say these are very portable skills, that the techniques we'll hear about today, the applications that we'll hear about today, you'll recognize how these too could be applicable in your own disciplines. So for the benefit of those watching from home or office or watching later in our broadcast series, I welcome you to the series. Please join me in welcoming Professor Christian Schonbach, who's the chair of the NU School of Science and Technology, Department of Biology. Thank you, Dr. Schonbach.
Thank you very much for the introduction. And I'm actually very happy to talk and being invited to this series. So today we cover bioinformatics and in various flavors as application to medicine, but also agriculture. So from the title image you can see that bioinformatics uses methods from statistics, mathematics, and it involves databases and programming. And so how I got into bioinformatics, I will introduce shortly. So you can find my profile on Google Scholar, then you get an idea what I published about and worked in. But I'm also currently serving as president of AP Bionet, which is Asia Pacific Bioinformatics Network, and we are engaged in education and organizing conferences. So my road to bioinformatics started actually in early 1990. When I did my doctoral thesis on orangutan, MHC, which is the equivalent to human leukocyte antigen. And we were looking at the evolution and organization of HLA, and the data available at that time were mostly like DNA, short DNA sequences. This was pre-genome era, and the reason why this is important is so if we look, okay, if you look at this slide on the right side, this scheme, you can see that the boxes represent variants of genes that in species X, some variant existed already, and then it was like transferred or inherited to species Y and Z. And we were collecting evidence for this to show that in this HLA system, this mode of evolution occurs, and then actually in 2012 it was also formally shown for the blood groups. So that on the left side you see species 1, this red box of variant B occurred already before the speciation. So that's why it's called trans-species polymorphism. In contrast to another mode called convergent evolution, where you get actually the variant A and B after speciation. So that involved not only wet lab experiments, but also computation. So at that time, issues were, unfortunately, the screen doesn't show that. Excuse me, can we have adjustment to the resolution because it's cut off? There's about 3 lines not shown. So at that time, retrieval was an issue, and also the organization of the, okay, just leave it. The organization of the data, because so internet just started to take off. At that time there was ARPANET, and transfer of data from databases was often not by internet, but you got an update of gene bank that where the sequences at time were deposited as a floppy disk or a CD by mail. So yeah, thank you very much. Good. Okay, so other issues at that time and still existing, so nomenclature and standards. So this is very important in bioinformatics, that we work with like high quality data and have proper controls, and that the community agrees on certain standards, including how to name things. Otherwise, retrieval becomes a nightmare, and also organizing the data in a database. Now, where does actually the word bioinformatics comes from, and what's the meaning? So, in the first line, I've put 3 words: so biology, information technology, mathematics, and highlighted in capital letters bioinformatics. So it consists of like these 3 areas, but there are various other definitions, and also depending where the people actually come from originally from which field, there are some variations. So I will show you 2. One is from Anna Tramontano. She passed away last year. So she's from Italy, worked at University of Rome as a biophysicist. And so she defined bioinformatics as science that uses the instruments of informatics to analyze biological data. Now, what are the instruments of informatics? So, for example, as from a user perspective, APIs, then we need some statistics, we need databases, we need programming languages. Then there's also hardware involved, so we need storage space and CPUs and analyze biological data. Now, biological data are quite diverse, and I will show various types of data later in the talk. And in order to formulate hypotheses about life. So bioinformatics is part of the knowledge discovery cycle, and now nowadays people often start out computationally and mine existing data and come up with some interesting associations, and then go to the wet lab and try to support those hypotheses. So another definition, which was published a bit earlier by Schonbach, it's more geared to he's a molecular biologist, more towards biology. So he defined it as conceptualizing biology in terms of molecules and applying informatics techniques derived from mass computer science statistics to understand and organize the information. So, and in to put it in short, bioinformatics as management information system. So he emphasizes also the importance of databases and data integration in order to utilize the information and, for example, come up with some hypothesis by mining those data in a smart way. And last not least, the practical application. It has many practical applications. So, for example, simple retrieval of sequence data, image analysis. When I look, for example, at microscopy data and want to extract information, for example, from such images. So, to give you a few examples. And the origins of bioinformatics, they date back, in my opinion, to the discovery of the DNA double helix in 1953. Watson and Crick published this famous paper of X-ray image of DNA in Nature, and then later they got the Nobel Prize for it. And on the right-hand side you see actually the schematic representation after analyzing this X-ray data. So to analyze this image, which looks a bit like a gray disk, so not only image analysis had to be performed, but also thermodynamics was involved, and this all actually relates to nowadays bioinformatics. The first time the term was used was published actually in 1953 by Ribac in French. That's on record the first time where the term was used, but it's interesting that he never defined it, he just used it, and in context actually of thermodynamics and also at that time when DNA sequences were available, the first short DNA sequences, he used that to write about and, for example, the information content of the basis of the DNA. So this is written here, and he mentions Shannon information theory here, and discusses about what is the maximum information of the DNA. And the same person, so Rybak, he designed also the first bioinformatics course in 1978. So interestingly, after that, you can't find in the literature much about bioinformatics in that like explicit way, and it took another like 15, 20 years until the preparations for human genome project began, and human genome project was in full swing, where bioinformatics again popped up and became enormously popular. Also, Queen Elizabeth in 1995, when she visited the head of bioinformatics of a lab in London, she complained it's a horrible word. So one can probably because it came from original Bioinformatique, which is French. Maybe they didn't go too well. Well, there's some important milestones I'd like to mention, which lead up to methods still in use today, and also resources. So in 1978, Dayhoff published a little booklet, and it was the first collection of protein sequences. So one can actually see this as the first database, but it was on paper, like a ledger. Now, this protein database still exists. It's PDB, and it's in electronic version. Then in 1986, Doolittle published book on sequence analysis, so how you can compare two sequences or multiple sequences. And below you see 1997, a name called ClassSal. So this is a program which aligns DNA sequences. It's still in use. And in 87, the first, what can you call AI, application was published in context of predicting the 3D structure from primary protein sequence, so just from letters, from the amino acids. And as Michael mentioned, so bioinformatics is intimately related with progress in technology and miniaturization. And sequencing technology is one of those disruptive technologies which enabled production of like lots of data and required then also bioinformatics to catch up analyzing this. And it started with in 1976. So Maxim and Gilbert published the first method of sequencing DNA that was pretty much low throughput by running shells on polyacrylamide shells and manually loading, and then waiting 1.5 hours until you get about 200 nucleotide readout. So this was later automated, and in 87 ABI, or Perkin-Elmers, produced the first sequencer, and automated sequencer. It's quite a large machine, and so that allowed to sequence, for example, phages, viruses, and bacteria. But still it was quite time intensive and also required a lot of manpower compared to the machine you see here. So ABI, this was the first generation of sequencer, and now we are, depending how you count, at the third or fourth generation, and this is one, actually, this little device, a little bit actually larger than the USB stick at my computer, is produced by Oxford Nanopore in 2009. You see these blue acronyms, PacBio, ON stands for Oxford Nanopore. So they produced this device, which is a mobile sequencer, and you can connect it to your laptop. So that can be used in the field, but also in the lab, and it allows actually to sequence an entire genome, human genome. So now, in 4 decades, you can see enormous progress driven by technology. And we don't have time to go into the details. So also, for example, the biochemistry of sequencing has changed. And now, Maxim Gilbert, so this was based on some biochemical method, and it required also amplification of DNA, whereas nowadays we don't need it. So we can go take a single cell and use some like fluorescent method to read out the bases. So the cost decreased and the data output increased, which is actually nicely shown by and recorded by NIH. So the cost per genome from 2001 until now, and was the Human Genome Project, the public as well as the private one, were enormously expensive. We are now down to about $1,000 to $2,000. And this is purely technology driven and depended on the engineering of new machines. So at the same time, with increase of performance, the output increased. We have in February 2018 1 billion sequences in GenBank or in GenBank, which includes also the whole genome sequences, and they constitute about 10 trillion bases. So that's, but compared to astronomy, it's still small data, but in biology, it's big data. And the red line represents, so WGS stands for whole genome sequences. And that started in 2002, it took off. So how do we get actually this there and extract from that double helix? How do we get actually these letters, the bases, A, G, T, C, the 4 bases of the DNA? So we skip the sequencing process and we start out with what these machines now, the 2nd and 3rd generation machines produce. These are shown as little red bars. These are so-called sequence reads. They're between like 20 to 30 nucleotides long. But now the human genome, is of course much, has many more bases. So how can we get from, for example, if a genome has 3 or 1 billion bases, how can we get entire genomic sequence? We need to arrange and find overlaps among all these small reads. So this is computationally very intensive. And there are also some problems involved in that, which relate to the composition of the sequence. We have letters A, G, T, C. So you have, for example, T, 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 and then A, A, T, T, T. So how do you know which of the T's comes first? So these are kind of trivial problems caused by repetitive sequences. Another problem is what if you have sequence error. You have, for example, N, you can't distinguish between the bases, or the base caller can't decide whether it's G or C, and provides like signal for both. So these are some issues. So I'll explain only in consideration of time. One case, what are we going to do if we have repeats? So the human genome consists of 50% of repeats. There's actually only a small part is protein coding. And we have about 1 million ALU repeats, about 300 base per long. And when it comes to finding out the order of these little fragments, so you can see they're actually interchangeable. So we need some trick to arrange this. Otherwise, it may happen that when we align our reads coming out from the sequencer, misaligned or misassembled as shown here, that we skip the part in the box in dark green. Because the two red boxes represent here repeats, and they were like misaligned and skipping actually this green part. So this happened initially when the sequence or the raw data analysis was not that advanced. And I showed two approaches that were taken, and now actually only one, which is a bit superior. But in principle, here graph theory is used. So graph theory is quite popular in bioinformatics, not only for making assemblies, but also networks in and pathway analysis in biology. So these are our reads. And if you use the Hamiltonian approach, so maybe some of you know this problem with the bridges in Königsberg. It's a mass problem. So this relates to this. And in this case, we connect actually trying to connect the vertices. And you see that we try to visit every vertex exactly once. And there's a red one, so you can see here was actually a problem. So this is one way actually to deal with these fragments and try to put them in the proper order to derive one consensus sequence. Another approach is Eulerian, where actually we're not looking at the vertices but at the edges. And in red, I've indicated one repeat, one type of repeat. For example, let's say 6 T's in a row. So we can actually do the same, and then in the Eulerian approach, because we use the edges, so we get actually a nice progression towards the the end, the yellow part of the sequences, and the repeats are shown, actually they're collapsed. So this is a Eulerian approach to this problem. And if you use that, also the algorithms usually doing this, they're linear in time. Whereas for a Hamiltonian approach, it's NP-complete problem. So nowadays, the sequence assemblers, they use actually Eulerian method, and with a twist. So the assemblers putting these small fragments together, they do actually, they take the Eulerian approach of the bridging graph. So I show now one example with multiple repeats just to show that this works. So we have different type of repeats, repeat 1 and 2 shown by different colors, and we can easily construct this or assemble the sequences with any number of repeats as shown here. And in the real world, so actually programs or assembler, they use the Eulerian tour of the bridging graph, and this is computationally, or when it started, was quite intensive because of memory. And considering that the human genome has 10 to the 10 times 1 billion edges, and so until 2012, SOAP was one, or SOAP de novo was one popular assembler, and 40 cores with more than 140 gig RAM required 40 hours to assemble the genome. So now actually we are down to hours, a few hours, by using Amazon Cloud and also a more memory efficient way of assembling. Now I want to talk a little bit about data types. So you've seen already the alphanumeric or the word version, using alphabet for the sequences. But to make this clear that bioinformatics involves also a lot of image processing, so I want to show this this figure here, which comes from one of the first generation sequences, and this represent the peaks for each nucleotide, and this because the bases the machine uses a laser, and then it measures the fluorescence. So and then the colors represent the different nucleotides. So this needs to be converted into letters, but also into numbers. And the numbers are used, for example, the peak height and the width as a quality measure. So there's in the second and third generation sequences, these numbers are quite important, and they're used, or the certain threshold are used to ensure that these fragments have good quality, and everything which falls below a certain threshold is actually not considered in analysis. There are other types of data, image data. They're more human understandable, so simple image data, for example, from cells or from microscopy, as well as microarray, where the user will see some dots in, for example, yellow or in red and green color. And other more demanding data, video data. So from microscopy, for example, live imaging that biologists follow the development of a cell, for example, over a couple of hours, see how it changes after, for example, inducing the cell with some growth factor. Then in behavioral studies, video data is used, and sound also is one data which comes out of behavioral studies and diagnostics. But I will show also one example, actually ultrasound, which is used in food quality control. Now all this data has to be somewhere stored after processing it in a human, sort of human readable form, but also in a like digital form suitable for fast processing. And I've listed here the like most popular biological databases. There's also actually a wiki site for list of biological databases. And I've talked about the NCBI, is quite in the middle, which contains GeneBank and hosts a couple of other databases. For example, PubMed contains literature data. Then there are a couple of others. PDB, for example, contains structure data, and so on. So an important issue with all this is that the community agrees on some standards, that the data can be integrated and is exchangeable. This is important for querying across databases, and also for like displaying or integration on the fly. So you can launch a query across, for example, 3 or 4 databases and merge these results and display to the user. And in terms of data sharing, so to make this public, most of these databases shown here, they are in the public domain, government funded, so all the data is open and for sharing. So when I mentioned the integration and exchange of data, so I want to show this typical workflow which supports this. This is quite common in bioinformatics analysis, that you make queries across different databases. And there's one example where you start out with a nucleotide sequence, then the user makes some gene prediction, and the red color shows the protein sequence. So then the user usually wants to know if this is, for example, some new sequence, what it is most similar to, and will query then, for example, UniProt-KB is a database which contains well annotated sequences, and then the user will get information about, for example, the function. Then if you want to dig deeper, then you need to know the 3D structure, and PDB is such a database where you can obtain protein structure. So in case there's no protein structure available, I can do modeling and look what is the most closely related sequence, and try to use then this sequence as a template to make a model of my new sequence. So this is one example of a workflow. Now one limitation is, although we have human genome and genomes of many other organisms, is actually this 3D structure. That this is a bottleneck also in drug discovery, because of 20,000 plus protein coding genes, only half of them have a structure. So there's still a lot of room for discovery. Another data integration type in bioinformatics is linking gene expression data with functional data coming, for example, from literature and pathways which were often manually curated. And such a resource is called linked omics, and it has about 1 million data points, 1 billion data points. So interestingly, this linked omics focuses only on 32 cancer types. So it's actually quite restricted, but it has that many data points. So this is just to give you an idea about what's with how many data you deal and how you integrate. So you can see, for example, these are text data. Then these are gene expression data, and they're visualized using heat map. This is a color scheme which tells whether a gene is highly expressed or low expressed. For example, red is usually high. And then, for example, the pathway enrichment method uses statistical methods to find out which of genes are overrepresented, usually shown in this red color, in a certain pathway. So, and what is shown here, this comes out of one query. So, and this is done on the fly. How can you do this? So you need some training, and there I will show 3 different flavors of bioinformaticians. So one is a bioinformatics user who has training in was trained to use some applications, and also understands actually the biology behind that. These persons mostly use interfaces. So, and the interfaces are usually built by the bioinformatics engineer. I will show in the next slide. And in between is the bioinformatics scientist. So the bioinformatics scientist knows scripting, can do some programming, and is well versed also in statistics, and analyzes the data. But so this person may not, bioinformatics scientist, may be not so good in, let's say, constructing some databases or optimizing it. This is a work of the bioinformatics engineer. So this kind of descriptions, they relate also to bioinformatics programs offered by universities, and depending where they are housed, so either computer science department or life science, they have different flavors. So this is the last one, the bioinformatics engineer. You can see that so they use prefer of course command line, and they develop, for example, he develops new data analysis algorithms, whereas the bioinformatics scientist more applies these algorithms. And there are also organizations which focus on training, not actually education in academic sense, but simply in training, professional development. And one is called Goblet. It's based, the secretariat is in the UK, but it operates internationally. And they are supported also by Elixir, which is a EU program. And they have a couple of every year bioinformatics events and workshops at low cost compared to commercial versions. So, and I was actually founding member of this, and we started this in 2012, and this is became actually quite successful resource, and especially for people interested in some specialized courses to use certain tools. And there are certain core competencies required, or people not required. Actually, there's societies in computational biology and bioinformatics came up with a position paper which was published in PLOS Computational Biology, where they suggest certain competencies. So this is quite useful document for example, universities planning to set up bioinformatics programs. And so another like more educational resource, encyclopedia, and Elsevier has in various areas encyclopedias in life sciences, for example, also in molecular biology, how you do certain conduct certain experiments. And in 3 years ago, we proposed encyclopedia for bioinformatics and computational biology. So this is going to be like sort of cookbook, and with some theoretical information included. And the first release will be this year in autumn, and there will be in 2019 the second release. So altogether about 300 to 400 chapters on all kind of topics. That's why it's also called Encyclopedia or the Bioinformatics from A to Z. And now I'd like to make a break before we move on to some use case examples in medicine and agriculture.
Okay, we'll pause here for a moment, but let me recap because I think Dr. Schonbach made some very important observations. Number one was the impact of the rapid growth of technology. You might recall from Dr. Varol's lecture when he described Moore's law and the impact on computational capabilities. And you saw from Dr. Schonbach the size of these devices that used to fill rooms, and now they're just a little bit larger than a USB drive, and they can attach to your laptop. So the evolution of these and the rapid growth of these devices has enabled tremendous progress. A second observation, the volumes of data are staggering, very large. They grow very large very quickly. And for us to conduct the analytics on this data, to help identify patterns, to look for anomalies, to detect disease, and so on, this requires enormous amounts of time and space when we think about computational resources. The other thing I think was very useful is when Dr. Schonbach was able to describe the skill sets required for the different kinds of professionals that you need to conduct this type of work. I think those profiles are very useful for young people who are looking to establish their careers, or for chief digital officers who are trying to identify what kind of people they need on their team in order to have a successful set of practitioners. Now, this would conclude more or less the technical part of the presentation, and as Christian has mentioned, after the break, in about 10 or 15 minutes, we'll look at some specific instances, manifestations, examples, use cases of bioinformatics, and we'll try to connect these to some current applications within the Digital Kazakhstan initiative. Okay, so thank you, Dr. Schonbach, and we'll reconvene in 10 or 15 minutes.
Можно один вопрос? И все-таки Почему выделен термин биоинформатика? Почему нет там медицинской информатики, химической информатики, ну и физической информатики? Почему?
I did some
My
translation.
question.
Huh?
Why bioinformatics? Why not
Ah.
informatics?
Yeah, it's a good question actually. I didn't explain this. There's also the term computational biology. And computational biology is usually used to describe the development of new algorithms and use mathematics to solve problems. This also not actually included in this profile for bioinformaticians because it's actually a different kind of job. So they're coming usually from mathematics. Why bioinformatics? As I try to explain this, and it's also, you will see this in the literature, bioinformatics doesn't have a clear, or the term doesn't have a clear definition. It's fluid. And depending who defines it, little variations. And the term itself, as you saw, comes from French, bioinformatique, and without any explanation actually. And Ribak wrote this, and when he tried to describe enzyme kinetics and the first application of information theory to DNA, I can only speculate about it because he died actually 3 years ago. That probably he thought about the combination information, science, and biology. That's my explanation.
Let's pause here. Yeah. Please, 10 minutes. We'll return for the examples. Thank you very much.
Polymorphism hypothesis actually doesn't go against Darwin.
Repeat please.
What I talked about, the trans-species polymorphism hypothesis does not go against Darwin.
Do you agree with Darwin evolution theory?
I agree mostly. There are some points I don't agree because nowadays there are other mechanisms, for example, epigenetics. Epigenetics is actually, you don't inherit. And what Darwin, 100 years ago, the people didn't know about this. So then they try to explain this with evolution. And nowadays it turns out it has nothing to do with evolution. But this is because they didn't know 100 years ago.
How 50% effects against opposite evolution theory. Opposite. 50% facts, yes. Opposites, opposite against Darwin's.
I know. Especially interestingly in America, North America, there are quite a number of people who don't believe in this.
I believe in telekinesis. Do you believe?
Well, I think
telekinesis.
I think it can. there's there's something to it.
Did you see these facts in India or some another place?
I read about it, yes, but
Did you see?
no, I didn't
No?
see it.
I'm sorry.
I didn't
Can I
see it.
interrupt?
Yeah.
I'm sorry, I don't have my English business
Okay,
card.
no problem.
I have a main question. You're teaching here some lectures to the students, right? In
Yes.
Azerbaijan University. I'm working for the Ministry of Healthcare.
Okay.
So we have right now like more than 14 terabytes of data of provided medical services to the population. Right now we're in the phase of personal decentralized data for the patient.
seems someone else booked the room at 3.
What's that?
I was told someone else booked the room at 3 o'clock.
We'll be done
Yeah.
in half an hour. We'll be done in
Yeah.
30 minutes. Right?
5 minutes late will be, now it's 25. Okay. Should
Okay.
be all right.
Okay. Okay, let's go ahead and get started for the use cases, the second half of today's presentation. In the interests of time, because there's another event scheduled after our session, we'll begin immediately.
Yeah.
So please, Dr. Schonbach.
So I got from the organizer of this event actually a wish list, and I tried to cover most of those points. And we start out with applications in medicine, particularly molecular medicine, and PPP, which stands for predictive, preventive, and personalized medicine. And again, I emphasize the importance of the data, and I'd like to start out with a fairly new resource. It's called the Human Cell Atlas, and this is an international consortium, Europe, Americas, and Asia, some Asian places, which started in 2016, and a few weeks ago actually the website went live. And this is not only a database, but also combined with biobanking. And the data stored include also actually patient data, and there are various levels of access to this. And why is this so important, the Human Cell Atlas? So the aim is actually to have about 10 billion data of 10 billion cells, and across all the human population. And this allows then to do, for example, genome-wide association studies, which in the past they were not always very predictive, or the outcome was ambiguous. This resource then can be used really to associate and certain genes with diseases, which can be used for diagnostics and also improvement of therapeutic approaches. And this is some like scheme where it can be used. So all kind actually was included also in your wish list. For example, in defining like people who do drug discovery. Once you have a compound, you need to test whether it's toxic. Then how efficient is in various populations. For diagnostics, then the effect of variants, everyone is different, how drugs actually act on individuals, and in regenerative medicine. Other applications here are shown in this workflow, which includes techniques I introduced, this various like next generation sequencing techniques together with bioinformatics workflows, and a couple of other biological high throughput and lower throughput methods to generate auxiliary data or confirm, for example, such sequence or expression-based data. And this is, if Kazakhstan is, I think, interested in this, so everyone, this is open, everyone can contribute to this. And there are other, some other strategies used in for bioinformatics towards personalized medicine. And one example is shown here. So common feature detection in cancer-associated pathways. And this is from a user perspective, where you have nice user interfaces. You can put in queries, and it pulls up genes associated with, for example, a certain cancer type, and also where the genes are located, or the gene products in pathways in the cell, for example. So this information is typically used if you have some cancer sample, was doctor requested sequencing of that, and in order, for example, to adjust some chemotherapy, and then one can compare what has been published already, or what effects of mutations in that particular gene have been reported. So before coming actually to this nice interface I showed and the database, one needs actually to do some work behind and come up actually with this information in order to generate these nice pathways and networks. This is one ongoing project I'm working on with a postdoc, and we're looking at a couple of proteins involved in DNA repair and cell cycle control. One is pCNA, and the question is, so which residues in this protein are actually co-evolving with interacting partners? And if you have somatic mutations in certain cancers, so how do they affect the function of this protein? So, and this is on the left-hand panel, you see co-evolving network of pCNA, which interacts with another protein called RFC1. And in purple, there's a residue, it's Q125, which seems to be fairly important, because you have many connections there. And there are in cancer database COSMIC samples from cancers where this position was mutated. Instead of Q, you have actually H125, and you can see the amino acid, so histidine changes the conformation. It's quite different. So this may affect actually the interaction of those pCNA with partner protein. And then one can go actually to pathways and look how let's say a less strong interaction may affect the pathway. And limitations are, again, it's a number of 3D structures and and also a number of samples, because one can always argue, how do you know that this is cancer, or how do you know that this mutation doesn't occur in normal population? And specifically when it comes to stratified samples, there's always a problem, for example, to get age-matched or sex-matched sufficient number of samples and controls in order to get statistical power. And so this is from a paper published some time ago by Valencia, who is actually head of the supercomputing unit in the Genome Institute in Barcelona, Spain. And he actually nicely listed here so principles of developing pipelines and key problems applicable and points to consider. So this is, I think, useful for planning such resources. And last part for this applications in medicine, I'd like to mention something. This is fairly new. This just came out end of last year. CRISPR, maybe some of you heard, CRISPR-Cas. This is now a really hot topic in gene therapy and any kind of modification of genes on a base level. So you can, with this system, CRISPR-Cas, one can change, for example, one nucleotide in a gene. And then this shows actually a system where this is combined with next generation sequencing, and one can actually sort of simultaneously see the effect of such mutation. And this is an example to as a principle of proof for the method where this has been applied to T-cell development, and they modified like 4 different markers for T-cell development, and they've shown actually the effect of the modifications on gene or transcript level and also on protein level. So to confirm that this is really true. And you can see here actually nice correlation between what is going on protein level and on gene level. So this is can be applied actually to in personalized medicine. For example, when you look at mutations in cancer or like response to drugs, so it has many applications. So. And the last one, it's it's not drug discovery was mentioned, but I'd like to make one example for vaccine development. There's a method called reverse vaccinology, which was actually pushed in the late 90s by Rapuli, who worked for Novartis, where you use actually, you start out with computational analysis. So this, of course, you need to know like your genome sequence of, for example, pathogens, but you need also the genome sequence of the host. And one can predict computationally epitopes, so which can recognize, for example, proteins of your pathogen of interest. And then based on that actually go through the typical pipeline of vaccine development, so animal study, then clinical trial until approval. And this is much faster than just the wet lab approach. And but also I'd like to make some reminders so that sometimes computational methods are really very fast. For example, apply artificial neural network or hidden Markov model to HIV sequences, and then predict which one bind to human leukocyte antigens. This is you can do for HIV in 15 minutes. You have the results, but then the problem is, are they really when you go to the wet lab, are those really epitopes? And if they are they effective? So a vaccine has to be effective. And so this first study, this was in 2002, but as you know, HIV, we still don't have a vaccine. And there are problems, for example, as shown here. This is a study, this was published, we published 2016, looking actually what happened when the virus tries to escape and you have a mutation in the epitope. Then you don't get a response, but why? And then it turned out, this was sort of accidental finding, that you can see here on the left side, this is wild type, and this on the left side upper corner. And this is an 8 amino acid epitope, and this is 10 wild type, and this is a mutant. And when you look at these red balls, so these are water molecules. Actually, what happened is in the mutated version, the number of water molecules fitting into this pocket changes, and then the hydrogen bond network changes, and that's why T-cell receptor doesn't recognize it anymore. So, and then you don't get an immune response. So these are sort of little findings which also you use computation, but slow down, for example, or reasons why a vaccine doesn't work. Now, I know that some use IBM Watson, and maybe you're aware of that. It was in the press last year. One reason was MD Anderson Cancer Center, because the way they implemented it was not very smart, and they paid a lot of money for it. But it works actually well for X-ray images, and but when it comes to more difficult problems, so finding gene variations, making associations, how course of the a disease or progress is affected by that, then it depends very much on how much actually curated data you have, and IBM Watson can actually train on. And so it's promising, but I think there are success stories, yeah, on smaller data sets, on problems, but I think it still takes some time. It's early stage. DeepMind was used in the UK to predict degenerative eye diseases and make eye scans for some people having diabetes, and this was actually from output was very good, but they mishandled data. And this was publicized in Nature. So when applying this kind of techniques, so then one needs to have also proper policies in place to avoid then a backlash by the public. So the next part will cover bioinformatics in agriculture and crop improvement, precision farming, bioremediation, and insect resistance. And one approach is microbiome analysis in two flavors. So I can look only at 16S RNA, which is fairly cheap, and because it's a short sequence you analyze, and then look how the taxonomic units or these sequence alignments actually split up. So this is a bit like phylogenetic analysis. So you get the different taxa of bacteria present in your sample. It doesn't tell you much about function. So for that you would do actually a shotgun metagenomic approach where you actually sequence in the entire genome. And this is just to give you an idea for the 16S. For this approach, this is really very quick. People usually look only at V3 and V4. And then you get a profile of bacteria present in your sample and also how abundant they are. And this allows actually to predict ecosystem functioning on various levels. So one can combine this with other data. For example, nitrogen content in soil, phosphor content, and or physiological data such as respiration, CO2, and these numbers, these are correlation coefficients. So with certain bacteria and actually which methods works well. So here they use two methods, biochemical method of phospholipid analysis plus the 16S RNA analysis. And you can see for some there is some anti-correlation. So it's for certain analysis it's good actually to use multiple methods, not just rely on one. One application, for example, using the 16S is what's the influence, for example, on the bacteria in soil without organic fertilizer and with organic fertilizer, in this case farmyard manure. And you can see after principal component analysis, so the different bacteria species nicely actually separate. And then one can, for example, if you can use this data and in, for example, adjusting organic fertilizer or adding, for example, looking at the soil, which what kind of soil, for example, comes closer to like the one like the blue FYM, the farmyard manure. So to have a good property of the soil, for example, selling to horticulture industry, for example. So another copper can be quite toxic, and that's why people are interested in measuring copper in the soil and also correlating this, for example, with bacteria. And again, you see actually can separate bacteria. Apparently, they can live with it or metabolize it, and others can't. So, and this is also done by this 16S RNA analysis. Another application is some certain bacteria are able to break down hydrocarbons, and to identify them, there's quite some interest going on in for bacteria living in the ocean to clean up oil spills such as Deepwater Horizon, but also leaks from drilling stations in the ocean. And Norway, for example, is quite active in that area. And these are unpublished data available in EBI metagenomics database. And they represent like various sequencing data of bacteria from like the ocean, actually floor, around Norwegian drilling stations. And a similar more detailed approach has been done after the Deep Horizon accident, oil spill, where as a 16S RNA analysis is not sufficient. You have to have the entire genome in order to identify then from certain bacteria. In this case, actually, they're focusing on this gamma proteobacterium and Colvellia species to identify exactly which genes and which pathways are important and are used by this bacteria in breakdown of certain hydrocarbons. And then one can, after obtaining this data, one can go back to the bench and do bioengineering to engineer, for example, strains which are able to clean up oil. In food science or in livestock, there's often integrative system genetics approaches are used, and the simple one is, for example, to make a cow with like lean meat or a lot of muscle, and this involves then either targeted sequencing or identification of QTLs. This is like related to like similar in human genome-wide association studies. In animals, it's of course a lot easier to do this. You have no ethical barriers are lower, and also it's much easier to split up between controls and individuals of interest. So this is used, so one is, for example, to improve the taste of meat, the texture of meat, in safety assessment, or also, well, biomass, for example, this is when, for example, manure is one example for that. So to use actually the excrements from farming, and another is like this important strain tracking. So this, for example, yogurt would be one example. What kind of lactobacillus is used in yogurt, so to trace food origin. And this is an example, I mentioned this earlier, about sound. Sound is also data used in bioinformatics, and this is also, it's an image, it's ultrasound. It's ultrasound image of live cattle. And it is used to assess the tenderness of like the meat between the ribs. And you want to do this, you want to know when is best time to slaughter the cow. And then you use ultrasound, and it turned out actually that now usually it's done manually, and but the ultrasound method is actually doing better than the experts, or if you do it manually and touch the cow. So this actually one can correlate this ultrasound images to percent of intramuscular fat, and if that is between 3 and 5%, then apparently the meat is very tender and tasty. So this is something very useful. And so another example is farming. So this is a kind of wild. It causes actually wilts and in rice. So, and they are testing actually how to improve insect resistance. And rather than using the rice, you use a model system which can be handled in the lab, which is Arabidopsis. And then use these results actually to, after the first screening, once you identified certain genes in Arabidopsis, then one goes actually to the plant. I'm sorry, it was not rice, it was cotton. And then go actually to the cotton plant in greenhouse, and then modify, for example, those genes in the cotton plant. So because the first approach is quite large scale, and one uses model organisms. This is similar if you do, when you test a drug, first you do it in mice. So this is a bit similar.
Dr. Schonbach,
Yeah.
if I may interrupt. In the interests of time, I understand there's another session that will begin at 3 o'clock here.
I finish soon.
Okay.
It's just one more. Because I feel it's important to understand this not as separate entities, because agriculture is also linked to health. And there is the concept of one medicine or one health, and I want to show actually example for that. It started in 1988 with Rappaport in Canada, who came up actually with this idea and founded an institute, and this was then taken up also by FAO and WHO. And so how that started is with actually antimicrobial resistance monitoring, and this is actually from the 70s. It was realized there's some connection between antibiotic resistance and the food chain. And now recently a paper was published actually on bacteria which bacteria occurring in soil and which show antibiotic resistant genes, and this is really scary. So this is from like Alaskan soils, and you can see the presence of bacteria. They are resistant to various types of beta-lactamases. So in some, so the bacteria are able to transfer this, and this is a real problem in the clinic. And this is the first time it was shown. There's a connection actually also to the soil. And this affects, of course, strategies to avoid antibiotic resistance in the hospitals. And this was the last slide.
Dr. Schonbach, thank you very much. That last slide was quite alarming. I understand why you wanted to mention it. Now, today was quite a busy day for many people, and some of our colleagues were unable to join us due to scheduling conflicts. So I would like to remind the people watching online and our colleagues who could not join that on Wednesday, Dr. Schonbach will run this seminar again in a webinar format across the entire country. And it's done in a more intimate way because he'll be narrating his slides, and we'll be using sequential translation for this rather than simultaneous. So for those of you who could not participate today, or perhaps you'd like to see some of this in greater detail, please remember Wednesday's session will be conducted at 3 o'clock. So we look forward to this. Also, I'll remind you that next Saturday we have another seminar which will be run on sensor networks and smart cities. So a completely different subject, but yet again another chapter in our discussions of digital Kazakhstan in our Center of Competencies work. So please join me again in thanking Dr. Schonbach. He's very busy running the labs and a department and an entire research program. He took the time out to participate today. So on behalf of our group, thank you very much. Okay. Until next time, thank you.